Good morning, everyone. Welcome back. I hope that you enjoyed your break from classes. Hope that you're having a good start to your day so far. Um, we have, let's see, we have just had a break for 1 week, and then before that, we had the asynchronous meetings. Um, I did get a couple of emails from people in one of my other classes that said that when they completed the ed puzzle, it said that their work was incomplete. But for those folks, I checked and I saw that they had a score, or they didn't have a score, I'm sorry, but it did indicate how many questions of the questions on their activities that they got right. And so what I'm going to do over the course of the next day or so is migrate those scores. Thank you very much. Thank you, Andrea. Um, I'll migrate those scores from Edpuzzle, and then if you have any issues or if you're like, hey, I did this, but this is the score that I got, please let me know. Um, and then I will take care of that. Now, with that in mind, I do have a picture of my son that my wife and I welcomed to our house, to our home. It's right here. That is that's our baby boy Hank. We're very excited about him. He's doing well, and so is my wife, which is great. So thank you guys very much for the kind messages that y'all sent. I really appreciate it. It's truly wonderful. Um, Now, with that, um, we have an exam on Wednesday, and I wanted to let you know of some changes that I'm going to make. And I'm hoping that it will make it, I guess, kind of easier for you guys or better for you guys. Thank you. Thank you. The first thing is that if you remember last time we did an exam, I had you log in on on your phone or something like that to Zoom, and then you used your computer to take the exam. Um, that has, I think that was really more of a burden than I wanted it to be. And it kind of made, I think, some of you guys' exam experience a little bit clunky. And, you're like, oh, I'm doing this and I'm doing this. Ugh. I don't want to do that again. I want you guys to have a exam experience that's a little bit more seamless. Um, so with that in mind, I am going to be on Zoom on our regularly scheduled class meeting. So that link. Um, in the event, and basically, I'm going to be there if you have any issues with Proctorio or you have a question about your exam, that's going to be your way to contact me. So I will be there. I'll be ready, waiting, anything like that. If you want to join through Zoom and join on Proctorio and take your exam on Proctorio, you're more than welcome to. Go for it. I will be here and I will be ready to answer any questions that you have or just, kind of be present while you're taking your exam. Um, but if you're like, I'm good, I'll just take the exam on Proctorio, then that's fine with me as well. Um, I just, yeah, I don't want you to kind of feel like burdened by oh, we have to set up our exams this way. I don't want the exam experience and the like technical aspects of the exam to influence or impact your performance on the exam at all. Um, I'd rather, your performance on the exam be based on your like knowledge of the material and the way that you kind of process that information. So that was the first announcement and first important part that I wanted to share with you guys. So I'm going to go ahead and blow up a PowerPoint though and kind of work through some things. Okay, so yes, indeed. I My pants are kind of green, so I'm going to count that as my contribution to St. Patrick's Day. Um, and I've got a green background, so that should count for something. Um, I hope you guys have fun. and go eat, what is it, a shepherd's pie and corned beef sandwich or something. Those are great. Though I'm kind of a quitter and I would just be like, oh, I'll just get a Reuben. Anyway, so today what I wanted to talk about was what you should expect for your exam on Wednesday. The exam, if you remember, all of our successive exams can replace preceding exam grades. So we've got exam number two. Your practice exam is up on Canvas now. Um, I might need to, I think it's available under the quizzes. I'm pretty sure it is. But I will send out an announcement sharing that with you. And I'll also send out an announcement with, details about the exam and the exam date and everything and reiterating like, hey, you don't have to join the Zoom. But if you run into problems, please do join the Zoom. That's going to be how we resolve it. Your exam number two, 40 questions. Can you use a note card? A 3x5, 3x5 card. Yes, but must be new and only one of them. And I clarify that just because I had a couple of people in my previous class who were like, oh, so we can use our exam one and our exam two cards? And I was like, nope, there's only one exam card. Um, so make sure that you kind of think about what you're going to put on there. Now, the next item is, well, what is this exam going to consist of? The exam is going to be 10 questions of your so-called old stuff. And if anyone is having an issue with the screen that I'm sharing, please send me a private message. I think that means I need to stop my share and restart it. 30 questions of the new stuff. Now, when it comes to the old stuff or the questions related to old stuff, um, I think that there's a kind of methodical and strategic way to um, figure out what you should study. Um, so when it comes to the old stuff, basically with our old stuff, we had, let's see, I'm looking back at my PowerPoint or my PowerPoints. Um, we had a couple of different topics. We had chemistry review. We had organic chemistry. We had functional groups. We had a discussion, well, two discussions on functional groups. And then after that, we had amino acids. So those are our four big topics for our old stuff. Now, when it comes to a question of like, well, how much depth are you going to go into on each one of these? And my response to that would be, I'm not going to go any greater depth than what was on your first exam. So what I would do is I would look at my old exam and try to pick out these different things. Um, what I'm going to do is, if you're thinking about, okay, well, how much time do I need to spend on these different topics? Um, I am looking at 10 questions. I'm probably going to go with about four for amino acids, two for functional groups, two for organic chemistry, and two for the chemistry review. And when it comes to um, amino acids, you have a couple of different things to think about. Classification. of amino acids. That was a big thing. Functional groups, identifying them. And same thing goes with amino acids. Classification, we had single letter codes, three-letter codes. So what I would do is I would make sure that I can identify, I didn't ask you to note every amino acid by their single letter code and three-letter code, but there are a couple. So I would make sure that I know those because those are going to be questions from your first exam that are kind of reworded, for lack of a better term. Um, so that's what I would sum up with my 10 questions from my old stuff. I would look at my review old exams, or not exams, exam. Review your old exam because that's what I'm going to do whenever it comes to writing your exam. I've also got the practice exam up, so look through those questions and try to figure out, okay, well, where does this come in? Um, but I give you these numbers because I don't want you to go into it with, okay, I have 2 hours to study. I'm going to spend 2 hours studying chemistry review and totally skip over um, amino acids. If you're like, I've got an hour to study, I'm going to spend close to 30 minutes on that amino acid stuff. Do I know my single letter code? Do I know my three-letter codes for the amino acids that are important for our class? Um, Now, the next thing for our new stuff, and if at any point you have a question or you want to, take a pause or anything like that, please don't hesitate to ask. New stuff. I'm going to kind of go through and highlight some of the important points regarding this. And I just got what I've got here is a slide deck that's basically everything compiled. And so I'm going to go through and kind of highlight different things that we talked about and use this. And all of the things that I highlight, I'll tell you, I'm probably going to highlight about 30 things at least. And those 30 things, think about those as, okay, this is a question that he might ask. Okay. So with that in mind, we talked about proteins this time. And we talked about the primary structure of proteins and amino acids. And this is a good definition. We had some good definitions here: primary structure, secondary, tertiary, quaternary. The primary structure of protein is the amino acid sequence from the N termini to C termini. So primary structure, N termini to C termini. How could I use that as a question? I could say, well, I could just give you the opposite. I could say the primary structure is from the C termini to the N termini. That's one thing that I could throw at you. Okay, the next level is secondary structure. Secondary structure is the way that the chain folds and coils. And all of this is related to the backbone of this polypeptide. Now, our two important structures are the beta sheets and the alpha helix. So how could I ask you about this? Well, one thing I could do is I could, do a screenshot, take this picture and say, this is an example of a primary, secondary, tertiary, quaternary structure. Or I could say, this is an example of an alpha helix or a beta sheet. In which case I'd want you to look at that and be like, oh, well, that kind of coils around itself. In addition to that, I could ask, well, what type of interaction stabilizes this? And that would be a hydrogen bond. Now, if I use this as a part of a question, I'm going to kind of block out the part that I have highlighted, that hydrogen bond there. But I wouldn't block this out right here. So that's kind of a tell. That's kind of a clue. Like, okay, we've got hydrogen going to oxygen. Aha, there's a hydrogen bond. Does anyone have any questions? Add to the chat if you think, okay, cool, I can see about 3 or 4 questions there. Sorry, total at this point in time. Okay, excellent, excellent. Um, okay, so now when we go on to, and if at any point you want me to stop or slow down or rephrase anything, please do not hesitate to ask me to. Next up, tertiary structure. I always think about tertiary and I think about three. This is the 3D, the three-dimensional shape of a protein. Okay, so the tertiary structure is your 3D shape of a protein. So that's the important part about it. And this is basically how, yeah, this is the good summary that you need. Now, what separates a tertiary structure from a quaternary structure is quaternary structure is two or more tertiary structures. So it's two or more things that have folded together. Now, a good question would be just the definition. The quaternary structure is it two or more? Is it three or more? Is it four or more? Or is it one, at least one? And the answer there is two or more. Now, another good question from that would be, well, a tetramer indicates how many tertiary structures, in which case you need to know your prefixes: di, tri, tetra, penta, hexa. If I went on from there, I'd go to hepta. If I went on from there, it'd be octo or octa. Um, so at this point, I think that I could probably come up with, maybe as many as 8 questions just from these first few slides. Um, After that, what are ways in which protein chains link to one another? Hydrogen bonds, that's a big one. Ionic bonds, disulfide bonds, and dispersion forces. Now, if you're like me and you're thinking, okay, well, how should I, is this important information? Yeah, it is. Is this information more important than my definition of a quaternary structure? It's not. So if you're thinking, what question is he more likely to ask? I'm more likely to ask to make sure that you understand what a quaternary structure is or a tertiary structure is than I am to ask about this. That's not to say that this is off the table, but if you if you don't understand the definitions of the quaternary, tertiary, secondary, and primary structures, but you can answer if you can fill this blank in, well, that's kind of just trivial information. And I'm not really about trivial information. Now, this is just kind of visual of those different interactions. So I might show you again, I might grab a screenshot of this and say, what type of interaction is this? And again, you're going to have hydrogen bonding, ionic bonding, disulfide, and dispersion forces all as multiple choices. Di, what does di indicate? Add it to the chat. Two. As soon as you see disulf, what should you think? Well, two sulfur. Perfect. Disulfide. Okay, two sulfurs. If I gave you this right here, that's got two sulfurs. Signed, sealed, delivered. Done. Okay. Now, that covers the important things about protein structure, the big things about protein structure. After protein structure, it's really important to know what the heck does a protein do? Now, proteins do a total of 3 major things. They're enzymes. They catalyze chemical reactions. They are involved in cell signaling and ligand binding. And then they're also structural proteins, so they give structure to things. I can move through this a little bit briskly, but here's our 3 different models. Enzyme catalysis. We've got an input molecule being split into 2 products. 2 different products result from this input molecule. So that's catalysis. Then we've got cell signaling. Here's a molecule that comes from outside of a cell to say, hey, membrane protein right here, stimulate some sort of interaction so that we get a response within the cell. If you remember, we used a kind of lengthy explanation as to cell signaling and ligand binding for, if you're, let me jump ahead a little bit. This was the example where I said, hey, it's like one of your friends is outside of the bar, you're standing at the doorway, and they're trying to get a message or they're trying to get a message to somebody who's inside the bar. Well, you play this little game of telephone. All these different players along the way do these small things to communicate the message until finally it gets to the person who is intended. So that's cell signaling. That's one of the things that proteins do. One of the other things that proteins do is they're structural players. So what that means is that they basically like maintain the structure of a cell. Keratins, for example, well, keratins are fibrous proteins. There's a bunch of these different things. Um, Fingernails, your hair, these are all examples of keratins. And they basically provide structure. Fibroin is a silk protein, a structural protein. It's got this organization, it's got a strength and an integrity to it. If you've ever heard about like spider webs and I think it's oysters on the side, like barnacles and forming barnacles, they get super strong. Um, and it's a protein that basically helps them adhere to the hull of a ship. Okay, next up is sugars or saccharides. So we've got monosaccharides. These are a base unit of sugars. Now, glucose was the one that we talked about as being kind of the most important, or I talked about that as being most important. There's different examples based on the number of carbons that it has. If a sugar has 3 carbons, it's a triose. 4 carbons, it's a tetrose. Pentose is 5 carbons, and hexose, a hexose, is 6 carbons. Glucose is an example of a hexose. So something can be a, or glucose, I'm sorry, is an example of a hexose. In addition to that, we also have to be thoughtful about functional groups. And this is a point where if you're thinking back to the what should I study for exam one material, this is a good thing to study. I'm going to go all the way back to the first slide that I had up here. Functional groups. You might be thinking, well, what's an important functional group that I should remember and recognize? Well, getting there in just a second. Okay. An aldehyde is a super important functional group. A ketone is also an important functional group. An aldehyde makes what's known as an aldose, and a ketone makes what's known as a ketose. So knowing and being able to identify those functional groups is really important. Glucose is known as an aldose. It is also a hexose. So these terms are they're not redundant because they mean different things, but they're redundant for glucose, for example. Um, Now, there are other aldoses. The only one that I really want you to know about is glucose. I want you to be able to identify this right here, that structure. So if I were to give you a structure and say, what is this molecule? Well, or, one other thing that I could do is say, this molecule or the molecule shown below is glucose, true or false. And what's important about this is knowing the organization of this molecule. So that's one question I might ask you. But more than likely, I'd ask something like glucose is an example of a pentose, hexose, octose, tetrose, alcohol. Or maybe not alcohol. I wouldn't ask that. None of the above. And I'd want you to say, oh, well, glucose is an example of a hexose. Okay, so then we talked about cyclization of sugars, and there's two different forms of glucose. But then more than anything else, I want you to know what are the components of sucrose, lactose, and maltose. Maltose is made up of glucose and glucose. Lactose is made up of glucose and galactose. Sucrose is made up of glucose and fructose. This is just like straight up definitions. So knowing those, if you kind of transcribe that onto your note card, you're going to get at least, two questions right out of that. Okay. Then I want you to know the type of bond. It's a glycosidic linkage that connects two sugars. Then let's see, where else do we have? Okay. Next up was glycolysis. Before I proceed into glycolysis, does anyone have any questions? Sure, absolutely. So it is a, I'll highlight it here. Two sugars are bound through a glycosidic, this says a glycosidic bond, but a glycosidic linkage is also an acceptable answer. And, if you're thinking, okay, well, what sort of question should I expect from that? Well, I would ask something as explicit as this type of bond connects sugars to make a disaccharide. And then you'd have answer option A, B, C, D. So let's say C would be glycosidic linkage. D would be, this is where I like to throw, I'll say hydrogen bond. Then I'll say, let's see, lophus scelic. And then binder. Okay, hydrogen bond is a real bond. Lophus scelic, I just made that up. And then binder is also not real. Can you repeat what slide should be on the note card? I took a picture of the last slide. Do you mean sugars, the disaccharides? Sucrose, lactose, and maltose. What are the individual sugars that make that up? Sucrose consists of glucose and fructose. Lactose is glucose and galactose. Maltose is glucose and glucose. Um, is this slide what you're looking for? So we have an aldose is an aldehyde, and a ketose is a ketone. Um, these are all good terms to also have on your note card, because those prefixes, anytime you see a number prefix or a numerical prefix and that OSE suffix, then you're talking about a sugar. Um, so I'll proceed from here. Yes, I will. So this is actually a compilation of everything. So I haven't posted this because this is just, I took all of the PowerPoints that we've covered for this module and I basically dropped the intro slide or the front page slide in the last one, and then I just compiled it all into one. So, but I will post this one because I'm going to save my notes on it. Um, so glycosidic linkage, then that's one of the ways that you'd see this sort of question about that. Then after that, we're going to glycolysis. Stanley, if you'd like to ask during office hours or something like that, I'm not exactly sure which one you're referencing. So maybe we could regroup on that. Um, but if I'll let me actually jump back, and when you see it, just say something to the chat, and I'd be glad to stop. So I presume that it is in sugars. Glucose is the one that I want you to know. Be able to like look at that and know what glucose is. Glucose is a hexose. It is also an aldose. Then here we've got these are the two different configurations of glucose as a ringed molecule. Then we'll just proceed from there. And then let's see. So that's a lot of information there. Then glycolysis. Glycolysis is basically the way that your body takes sugar and breaks it down and gets energy from it. So I want you to know what is the input and what is the output. This is our input. This is a form of glucose. And then this is our output. Now, when you look at this right here, I always like to just kind of take a very basic approach. And I like to think glucose is 6 carbons. Okay, and then pyruvate, our output molecule, is 3 carbons. 1, 2, 3. So if we take a 6 carbon molecule, if we take one 6 carbon molecule and split it, we're going to be able to split it into two 3 carbon molecules. So you can kind of think about glycolysis as we're taking a 6 carbon molecule and we're basically breaking it in two. That's one approach that you can take. Now, a follow-up to that is that all there is to it? And the answer is no, there's more to it. So there's more to it is kind of our overview. We're investing ATP, inorganic phosphate, ADP, and NAD+. We're returning ATP and NADH. Now, this is the big overview of glycolysis. What do I want you guys to know about the big overview of glycolysis? Is I want you to know about glucose, the input, hexokinase, our first enzyme, phosphofructokinase, our second enzyme, or our second irreversible enzyme, and then our last irreversible enzyme known as pyruvate kinase. Um, and then last but not least, so those are our three irreversible enzymes. We're beginning with glucose. We're investing 2 ATP. We're ending with glyceraldehyde 3-phosphate. This is known as our investment phase. So I want you to know what our investment is and where we can go from there. And then I want you to know what your investment phase starts with and what it ends with, and what is done along the way. Well, you've invested 2 ATP in the process. Now, your return phase. The return phase begins with glyceraldehyde 3-phosphate and ends, and I'm going to go ahead and add in here 2 glyceraldehyde 3-phosphate, and ends with 2 pyruvate molecules. What are we getting out of this? We're returning 4 ATP. We invested 2 ATP, we return 4 ATP. So our net ATP is 4. Or sorry, our net ATP is 2. Um, There are three big enzymes that I want you to know: hexokinase, phosphofructokinase, pyruvate kinase. Hexokinase is irreversible. Phosphofructokinase is irreversible. Pyruvate kinase is irreversible. Phosphofructokinase is the first committed step of glycolysis. That's basically saying we're starting along this path and we're going to finish this path. Hexokinase is not committed to glycolysis. It's committed to making glucose 6-phosphate, but it's not committed to glycolysis. That's an important distinction that I want you guys to know about. Okay, so that is one of the big deals with ATP and glycolysis. Now, next up, DNA. So if you're thinking, before I proceed into this, if you're thinking, okay, well, those were all of the things that we did synchronously, that we had class meetings for, and then the remaining two slideshows that I'm going to go over, that's stuff that was done asynchronously. Um, I'm going to have a little bit more of the questions related to the synchronous stuff. So if you're thinking, how am I going to split my time? I would say 60 to 65% of your time should be spent on, no, my numbers are a little bit off. Most of your questions are going to come from up until this point. Will you be asked questions about DNA and RNA? Absolutely. But you shouldn't expect 35 questions related to DNA and RNA. Okay. Now, there's a couple of important experiments. Hershey-Chase experiment. What was the significance of this? It determined that DNA was the carrier of genetic information. The DNA double helix is the structure that was determined with the help of Rosalind Franklin. She's the woman here on the far left here. Her work was instrumental for the people who were ultimately credited and awarded the Nobel Prize. Without her work, they wouldn't have been able to resolve the structure. Or they would have, I think, gotten there, but it would have taken considerably longer, which would have delayed everything. Now, last but not least, the Human Genome Project. What is the human, or what is the sequence of the genome? That was J. Craig Ventnor and his group who helped sequence all of the genes in a human genome. Okay, so DNA and RNA, deoxyribonucleic acid, ribonucleic acid. Sugar. Well, we have a sugar molecule as a part of a ribonucleic acid. Our sugar is ribose. Deoxyribose, we've got a deoxyribose. Now here what we have, one important thing is the 5' and 3' carbons. Do we have to remember the people's names? I would remember Rosalind Franklin and the Hershey. I would remember the names of Rosalind Franklin. What was the significance of her work? I would remember Craig Ventnor as human genome, and I would remember Hershey-Chase experiment. Those are the three things that I would remember. So yes. Um, so our 5' to 3' direction, that's our primary structure. We read DNA in the 5' to the 3' end. Same thing with RNA. We read it in the 5' to 3' end. These polymers are linked by what's known as a phosphodiester bridge or a phosphodiester linkage. And they make up ribonucleic acids and deoxyribonucleic acids. So that's an important thing to pull away from here. 5' to 3' directionality. Then I'm going to move on from there. Sugar phosphate backbone. That gives DNA an overall, like a DNA molecule, a negative charge. Then, hydrogen bonds are what hold the two strands together. And then a nucleotide is a nitrogenous base, a sugar. and a phosphate group. So this is a question that if I highlighted this, if I said, hey, what is this? Versus if I said, what is this? What is in box A versus box B? Box A is a nucleotide because it has a phosphate group, sorry, a sugar right here, and a nitrogenous base right here. Nucleosides, by comparison, well, that's what's in box B because that doesn't include that phosphate group. So small difference, but important difference. Um, then ATP, ADP, AMP, well, that is referencing adenosine triphosphate, diphosphate, monophosphate. Nomenclature related to what just happened. Oh, feel like something's happening. Everything's running slow. Okay, there we go. Okay. I want you to know how many bonds are between different nitrogenous bases. AT is going to be two bonds, two H bonds. GC is three H bonds. So I always like to think about this as which is stronger, the linkage between A and T and G and C, or G and C. G and C has three hydrogen bonds. A and T has two hydrogen bonds. Now, last but not least is lipids. Lipids are biological molecules that are soluble in organic solvents. This is very important. This is important because most everything in your body is not an organic solvent. And so these things kind of like, it's like if you poured gasoline into a glass of water or something like that, that gasoline is going to kind of separate itself from the water. Now, lipids have three big functions. They are, they make up membranes, they are energy stores, and they involve signaling. Major classes of lipids, fatty acids, triacylglycerols, and steroids. We focused on fatty acids and we looked at these guys right here. We had saturated versus unsaturated fatty acids. This right here is your saturated fatty acid. Because in the carbon or the hydrocarbon chain, there are no double bonds. All these three, by comparison, are unsaturated. I did ask you to know about the notation. Like if you saw, go ahead and answer the in the chat. If I gave you something like this, C16:4, and I asked you the question, first question is this an example of a saturated or unsaturated fatty acid? Add to the S for saturated, U for unsaturated. Add to the chat. Perfect. Unsaturated. Now, my next question is, how many carbons does this fatty acid have? You got two numbers to work with, so it's kind of 50-50. 16 carbons, perfect. How many double bonds does it have? Well, four. You guys all answered that correctly. I've posed that question in the past, and that's where people go and they kind of get a little funky and they're like, ooh, 16:4. I'm going to say it's 12. Don't do that. There's two numbers. The first number says how many carbons you got. The next number says how many double bonds you got. Can I get a thumbs up or a yeah if we're good with that in the chat? Thank you. And I'm pausing here because should you expect this on your exam? Yeah, yeah, yeah. One of those questions just like that. Okay. Next up is we talked about omega fatty acids. Omega fatty acids are found in like salmon and eggs, and fish in general, I'm sorry. Now, the significance of fatty acids, I talked about this a little bit with respect to foods. So, saturated fatty acids generally, I think the nutritional advice is to not really consume so many saturated fatty acids. And the reason is because they pack really tightly. And the significance of that is when they pack really tightly, that just means there's lots of energy stored there. And, if you're someone who gets up or if you like ride your bike, I'm just going to use cardio as an example of an activity because I know that a little bit better in terms of like caloric expenditure. Um, or you could say, if you have a job where you're working out in the sun 12 hours a day, and then when you get home, actually, heck, I'll use this. Has anyone ever heard of David Goggins? If you haven't, he's pretty cool. Look him up. He's an interesting and inspirational kind of guy. Um, that's right. That's right. Oh, he okay. Yeah, yeah. Um, yeah, I really like him. He swears a lot. That's kind of funny. Um, and yes, he bullies me on TikTok. That's absolutely right. Every time I stumble upon like a YouTube clip or an Instagram clip, I'm like, man, I need to I need to get outside. I need to do something. What do I need to do? I don't know. I feel like I need to build a brick wall or something like that. Anyway, my point is that David Goggins, he runs like I think he runs like 14 miles a day in addition to just having like I heard a couple of years ago he was like working as a firefighter in the redwood forest in California. Um, so he's an intense dude. He burns lots of calories just by working. He needs to eat a bunch of saturated fats. Um, so he needs that energy. Um, I'm sitting right now on my computer. David Goggins would be yelling at me right now that I should be doing push-ups or pull-ups while I'm at my computer, if anything. Um, so I don't need as many saturated fatty acids as him. So anywho, that's the big difference there. So saturated fats, they pack really well together and then they kind of organize and there's a lot of energy there. Now, with that in mind, that's one of the important difference between saturated and unsaturated fatty acids. They pack really well together. Unsaturated don't pack very well. Now, here what we have are examples of fatty acids and some of the properties. I mentioned this one as relating their melting points to the number of carbons. What do you see? What's a trend as the number of carbons increases? What happens to the melting point of these? It goes up. Now, when you drop down here, we've got unsaturated fatty acids. Well, there's two things going on. So it's a little bit complicated. We've got number of carbons that are increasing, but we've also got the presence of double bonds. 18 colon 3, three double bonds. 18 colon 1 versus 18 colon 3. Our melting point is 13.2 and negative 17. As your number of double bonds increases, what happens to your number or your melting point? It decreases. So you gotta like be ready to answer a question about that. As the number of carbons increases, what happens to the melting point? As the number of double bonds increases, what happens to the melting point? It's complicated whenever you have multiple things going on, but I want you to be able to answer those specific questions. As number of carbons increases, as number of double bonds increases. I'm not going to throw something, well, what happens when both things happen? Well, then you kind of have to like prioritize them. But that's not a part of what I'm going to be asking you guys. Okay, so there we go. There's our examples. Triacylglycerols are the kinds of fats that um, make up like membranes, and they're a big part of membranes. They have two or three different fatty acids roles that they play. Well, They serve as energy reservoirs, and that allows basically sustained starvation. And if you're fasting or something like that, if you can fast for a month or something like that, and your body's going to go through and start breaking down triacylglycerols and getting a lot of energy from that. Um, there's an enzymatic process where fatty triacylglycerols are broken down, and then there's an industrial process known as saponification. So this is done in industry. If you've ever seen the movie Fight Club, that's what's happening here. And then enzymatic hydrolysis is what happens in your body. Cholesterol is an example of a steroid. It's not exactly the most exciting. Whenever you eat a bag of Doritos, you're not ingesting a like anabolic steroid like Mark McGuire or Jose Canseco or I don't know who any other recent baseball players who have violated performance enhancing rules, drug rules, but cholesterol, that's what cholesterol looks like. And this is going to do lots of different things to your body. There's steroid hormones that go back to what we were talking about of signaling. Testosterone is an example, prednisone, aldosterone, cortisol. These are all hormones that regulate basically functions within your body. Um, then we talked, or I mentioned vitamin D. It's involved in calcium and phosphorus metabolism. What I want you to know about vitamin D is that you got to have a good functioning liver and a good functioning kidney in order to make um, vitamin D3. Now, if you take a vitamin D3 supplement or a vitamin D supplement, this is the stuff that you're getting. So in the event that somebody's having trouble with their liver or kidney or, deteriorating function or something like that, they can take a vitamin D supplement and don't have to process it at all or anything like that. But those are the big things that I want you guys to know about. This is a lot of information. What I would say is that for the DNA and lipid stuff, it's on your exam, but it's not equally as weighted as all of the other stuff. Now, I'm going to go ahead and stop my share. If you guys have any questions, I'll hang around to answer some of those questions. If you got to go and get bullied by David Goggins to go for your daily 14-mile ruck run, do it. That's what Goggins wants. But thank you guys very much. I think I got to cancel my office hours today because one of my kids is sick. I got to take him to the doctor. But if you got questions, I'll be around here for just a little bit if you got quick questions. Other than that, though, have a great day. I'm going to stop my recording and I will talk to you later. Deal?